I'm just going to say hello to those people. It is now 4 o'clock, so the web class has officially started. Let's see how this is going to go. Where is my... Can you guys still see me? I've lost my chat temporarily. There it is. Boom. Okay. Started. Let's see how this is going to go. Where is my... Can you guys still see me? I've lost my chat temporarily. How do I mute this? There we go. Boom. I'm just hearing myself on loop. You can see me. Right, okay, 4 o'clock, let's crack in. So let's go through some of those competitions. I'm going to show you effectively where you can interact with us this week and how you can get involved. So there is going to be loads of stuff happening on YouTube, loads of social media happening on Instagram. So without further ado, let me share my screen with you so I can show you exactly what's going on and how you can basically win some stuff. Free stuff. Everybody loves free stuff. Okay, so one of the competitions right now is basically going to be for tagging a friend on one of our Instagram posts. In fact, I think we're going to update the post a little bit later on. I'm going to come back to this one. But basically, if you tag a friend and follow the Instagram page this week, then you'll be entered into competition with someone at the end of the week will win it live. So yeah, you will on the Sunday web class, where we're going to announce the winners to all of the competitions that are running all week. So essentially, what you'll do at some point this week, you've got to follow the Instagram page, tag one of your A-level biology, chemistry, or maths friends in it, and we will pick a winner by the end of the week on Sunday. The web class is at 6 p.m. And yeah, we'll announce all the winners there. You've got to be in the web class in order to claim your spot. So if you're not there, you're not present, you don't get to win. So we'll pick a few winners, and just in case a few people aren't present. Okay, other ways that you can win this week are to basically leave a review, a public review on the Facebook page. It's got to be public, because if I can't see it, then I can't pick you as a winner. It is that straightforward. People have in the past said, well, I left a review, where was mine? Didn't show up on the page. It's because you didn't leave it as a public review. You're only sharing it with your friends, then you're not. Okay, so what else can I say to you? Let's kill that. Okay, let's go. So leave a public review. The winner is chosen totally at random. You can review it however you like. However, yeah, anyone between, well, this was the last review done on the 16th of April, ages ago. So basically anyone reviewing Tailored Tutors between now and Sunday, again, the winner will be announced in the Sunday web class. What else can I go through with you? What are some other free things? If you're going to stay to the end, I'm going to show you how you can basically get an unlimited all-access pass to the biology, the chemistry, the maths, absolutely unlimited pass from 8 a.m. Saturday until 6 p.m. on Sunday. You can access all of our biology and chemistry and maths video revision tutorials absolutely free. I'm going to show you how to do that at the end. So just basically stay tuned to the end, and you will be able to figure out how that's, all you got to do is bring a friend. I think for now, I'll just go over the schedules just so you guys can actually see what's going on this week. So we are here, all the AS biology answers in 1 hour, starting at 4 p.m. 6 o'clock, we've got, I don't know exactly what the title of that, Rich is actually going to do a few different bits and pieces in his chemistry web class. So the chemistry web class has been tweaked slightly, and then we do math C1 and some AS Pure stuff going on at 8 o'clock with Ronnie. And then tomorrow, Wednesday, we've got chemistry and maths, 6 p.m., 8 p.m. Thursday, chemistry and biology, 6 p.m. and 8 p.m. Friday, maths and biology, 6 p.m. and 8 p.m. And then the weekend, we've got an all-round web class for any student of any subject. So you're welcome to tell your friends even if they don't do these subjects. You've got how to improve 2 grades without doing more work. And without doing more work, I mean without doing extra work. You can use the amount of time that you should be working, like a regular amount of work, without piling on loads of exam pressure, and actually tweak your grades to get better grades at the end of it. It's going to be how to use a lot of combination of stuff. And then we're going to do a drop-in Q&A, any questions you've got from any of these kind of subjects, starting on Sunday at 6 p.m. So once your all-access pass is going to expire at 6 p.m., you can jump on the web class and ask us any questions you've got about it there and then. Everyone happy? Everyone stoked? Everyone excited? Can you get a recording? Yeah, I'm going to cover the recording. So remember that interaction pass. Those, oh, in fact, I didn't mention that if anyone's joined us just after 4 o'clock, those people interacting with us live, basically, you are going to be able to, those people who are interacting with us the most, answering my questions as much, asking intelligent questions, or like questions as opposed to just writing any old questions in the chat box. We are going to pick someone at the end of today, and they're going to win it absolutely free. You're going to win access to the course of your choice at the end of the week, whether that's biology, whether that's chemistry, whether that's maths, you can choose. But the person who is most engaged in today's web class, we're going to give one away for free. Okay, are you ready to get started? This exam, this is going to go through AQA, it's going to be Edexcel, it's going to be OCR. So this is going to be general to all of you guys. I've picked topics which are obviously suitable to all of you. Boom, boom, boom. Okay, I'm just going to throw a poll at you just so you've got, I know that you're not asleep. Which poll do I want to do? I want to know, have you used any of our Tailored Tutors videos before in the past? Which ones of you guys, how many of you are completely fresh? How many of you are like, you know what we're all about? You know, I don't have to spend so much time prepping you guys. Maths. Yeah, the maths course is awesome. The chemistry course is great too. Okay, so actually I'm getting a bit of a split. I like at least 80% of you guys to vote, just otherwise I want it to be a nice representative survey. There's now well over 300 people in the room, so we should get a nice large data set. 80%, we've hit it. The faster you guys can do it, the faster I can share it with you. Boom, so 60, well, okay, 2/3, 1/3, pretty much bang on. So the 2/3 of you, I'll be giving you a little sneaky peek, a little bit of a view inside the course today as well. Deep breaths. Let's go. Okay, it's to the presentation. Although I'm not at the beginning of the presentation because I was just checking it through. Let's go to the beginning. It's been a busy day for me. Any students in there from Featherstone High School? I was at Featherstone High School this morning in London. It was, yeah, I just shot back home and hastily why this presentation isn't quite done because I was pretty late getting home. Okay, so recording is recording. The recording will be posted where you click the link in this live web class. That is where this recording is going to be posted. So make sure that you are checking that out. And yeah, it's going to be there. It's going to be fast and furious. The next, however many, 57 minutes plus Q&A. It's going to be really quick. So yeah, you might not be able to. What's going on? What's going on? Get your preparation. Okay, so yeah, it's going to be super quick. The recording will be there until the end of the week at the very least for you to go back and take some notes and scribble it down. I suggest you try and keep up with as much as you can. Obviously, you're going to be typing in the chat box. So now it should zoom into 100%. Genius. Okay, so the chat and the Q&A. So the chat can just bubble along. I'm going to save the Q&A basically when the end, or if I've got some specific questions, I will tell you to go to the Q&A and sort your Q&A out, and you can submit some questions there. Okay, so without further ado, let's actually get stuck into some biology, if I can. Okay, moving across. So these are all like super condensed summaries of what you are likely to get asked, things that come up, the details that you really need to know. And we're going to maybe look at some application style questions later in a little bit as well. Okay, so if we get questions like this question come up, it won't come up exactly like this. They're not going to be a fill in the table, but these tables are a great way of testing your knowledge with some active recall to figure out whether you know it well enough or whether you don't. Okay, so let's say, what are the two monosaccharides that make up maltase? So we've got, well, glucose is in all of the monosaccharides that you need to know about. So let's stick glucose in down here. Glucose plus what equals maltose? Well, it's actually pretty straightforward, the maltose one, because it's just glucose and glucose. Okay, that leaves us with two question marks. Lactose has a little bit of a clue in there. If you're, again, anyone raising their hand, I'm sorry, I'm just got no chance. You can see how quickly that chat's zipping along. I've just got no chance. You can stop doing that right now. Otherwise, you ain't going to win that interaction prize because it's going to annoy me. Okay, so glucose and lactose is going to be galactose. I get a record of that as well. Okay, so next up, we're going to go glucose and what makes sucrose? You got it, fructose. Okay, so now, like this kind of information, if it's not fresh in your head, especially if you did this 2 years ago, then this is going to be some like really quick recap. So these are the kinds of questions that they're going to want us to answer. Like we probably are not going to have to draw alpha and beta glucose, but if we do, how's it going to look? Well, we've got the oxygen, so let's not forget the oxygen in our hexose. And that means we've got, if we number our carbon atoms, 1, 2, 3, 4. This is an oxygen, it's not a carbon. This is 5, which means we have to add a 6th one. Don't forget the 6th carbon up here. And with alpha glucose, then we're going to go OH is here on the bottom. In my video notes here, I give you nice ways of remembering these. So I think say Santa ho 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 comes down the chimney, and you basically get these coming like the OHs are on the bottom for alpha, and Santa has alphas in it. Okay, so beta glucose is going to vary. We're going to have the oxygen there, so we can draw the basic structure exactly like this first. And we can have this side being the same, but on the other side, we've got our OH flipped up the other way. So this is our beta glucose. And if you can't just sit down and just sketch them out off the top of your head, then this is something you probably need to work on. So the bonds between carbohydrates is glycosidic bonds. And they always, if you get a suggest question, suggest what happens between the bond between these different molecules, well, we're probably going to get a glycosidic bond. If it's carbohydrate, it might be a crazy sugar you have never heard of before. But if it's an OS, if it's a sugar, if it's a carbohydrate, it's going to be a glycosidic bond. It doesn't matter if it's a describe or explain or state, it will be, if it's carbohydrates, when you join them together, it's a condensation reaction, you release a molecule of water and it's a glycosidic bond. Okay, polymers of alpha glucose. Well, alpha glucose, we've got a few. We've got starch, but starch is actually a mixture of two compounds. We've got amylose, and we've got amylopectin. And then we've got glycogen. These all have the same function, and that is to store glucose. I'm going to come on to that in a minute. And the beta glucose, the polymer of beta glucose is indeed cellulose. Moving on. Okay, so again, you can see how I kind of laid out in my notes. So this is my little annotations of Santa down here. Santa is got A's in it for Anta, and Santa says ho, ho, ho. He also comes down the chimney, which is something I say in the video, but I don't actually draw it on there. So that's how I remember. It's stupid. It doesn't actually make that much sense, but it certainly gives you a little, what I call a memory hook, something to think back on. You're going to build your series of notes. It's all going to be color-coded. It's going to be super memorable, maybe way more than so than reading a shutter. Okay, so summarizing the polymers of glucose and how they're going to be useful in organization. Okay, so let's rip through this table. What's the function of cellulose? Function of cellulose is going to be structural, so cellulose cell wall. So I'm going to put structural slash, and these are all based off past paper questions, cell wall. Okay, let's go across. Whether we can go across and whether we go down, it totally doesn't make too much difference to us because we've got to know these, and we've got to, again, these kind of facts have got to be pretty quick recall for us, because when we get to those like application style questions, we need to spend a bit of time thinking about those. So if we can get through those simpler questions as quickly as we can, I'm going to go across for some and go down for others. I'm going to mix it up. I'm not even going to complete this table. There's just not going to be enough time. There's so much content in this web class. So amylose, amylopectin, and glycogen. Well, I'm actually just going to do this in here, and this is glucose storage. And it's the same for these three. Okay, type of glucose, beta, alpha, alpha, alpha. My chat window is floating above the glycogen column right now, so I can't see that too easily. Bonds. Okay, so what type of bonding? When I numbered my carbon atoms previously when I drew it out, we've got an oxygen here. We've got, this one is considered carbon atom number 1, number 2, number 3, number 4. This one isn't 1, remember, this is number 5, and this is number 6. Okay, so we're going to make bonds with this. We got straight chains. Cellulose forms straight chains. So it's carbon atom number 1 with carbon atom number 4, which is why this is called 1,4 linkages or 1,4 bonds. So anything that is unbranched, such as amylose, is going to have 1,4. They're all actually going to have some 1,4. These are like the staple bonding between glucoses. But to branch it, we need a T-junction. And to get that T-junction, it's going to come off number 6 as well. So this is anything that's branched. Which ones of these are branched? So we've got yes, no, yes, no, yes, no. And I'm going to put the only difference I'm going to put in this column between this one and amylopectin is very branched. The only real difference, obviously, we could put animals in here. And this is obviously in plants. So that means we've got 1,4 bonds and we've got 1,6 bonds. Or linkages. You can call them either. You're not going to get marked down for either of those in the exam. Okay, and so this is therefore unbranched. Okay, soluble, insoluble. Cellulose cell walls wouldn't be that much use to us if they dissolved in the rain. So cellulose is kind of self-explanatory. It's insoluble. Okay, what the questions that they might ask, they never ask about cellulose being soluble or not, but they will ask about amylose and amylopectin or glycogen being soluble. What is the benefit to amylose, amylopectin, and glycogen being insoluble? Why would it be a bad idea? Why don't we store glucose as glucose? Bang on, zipping through the answers. There's like over 400 people in the room right now. Okay, so do get involved with those comments. I can see them flying through. It doesn't affect the water potential. If we store glucose as glucose inside it, especially an animal cell, then what's going to happen? The water potential goes down, 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 down. We're basically making super concentrated glucose solution. Then all the water wants to do is come in by osmosis. Animal cells don't have a cell wall. That cell will burst, or lyse is the technical term, L-Y-S-E. So they're all insoluble. And can we sketch the structure? Well, we can sketch the structure. So we've got beta glucose because it's got that flipped oxygen, because it's got the OHs on one side and the other. When we bond a 1,4 linkage in a carbohydrate, we actually don't get a perfectly straight line. An ester bond is a pretty straight line, but what we get when we bond in a glycosidic bond is we get a kink. But in beta glucose, we go kink one way, kink the other, kink one way, kink the other, kink one way, kink the other. So we end up with these straight microfibrils that are joined together by hydrogen bonds. So they're unbranched and they're straight chain, and they're super strong because there's loads and loads of parallel chains, microfibrils, held together by hydrogen bonds. When you get amylose, it's not, because it's made of alpha glucose, it's kinking in the same direction each time. So we get this, and then basically what do we get? We end up with a spiral. And amylopectin is going to be exactly the same. We're going to have this spiral, except because it's got some 1,6 bonds in it, it's going to have spirals coming off it like this. And that's why we get branched. And the only difference, as I said, this amylopectin and glycogen is exactly the same content, except glycogen is glucose storage in animals, and it's more branched. Why is it more branched? What's the advantage of having branching in amylopectin and glycogen? Okay, so it has a greater surface area. Or basically, when we snap off a glucose, where are we going to? Are we going to go taking a glucose from here, if this is a polymer? No, we're going to take basically, it's got more ends exposed. If we want a glucose, we're going to take it off the end, right? If you've got a long string of sausages, let's say, all strung up in a row, I'm just making this up as I go along. If you want one sausage, does it make more sense to take it right out the middle or take one off the end? Take one off the end, of course. And therefore, the more ends you've got, the more accessible you've got glucose. And therefore, it's got a bigger surface area for the enzyme. What's the enzyme that breaks down starch called? Amylase. And why is it called amylase? Well, why is it not called starch age? Because starch isn't really a thing. Amylose and amylopectin is the enzyme which is going to break down starch. Boom, boom, boom. Okay, so again, how does this look inside the course? Well, it looks pretty much the same as that, but you've got a full explanation. You go through what the function and the structure of each of these things are. You get more diagrams. You basically get all the little bits of detail. You get all the proper notes. Obviously, I am going at breakneck speed right now. These videos are all going to be 6 to 10 minutes typically, and they're going to break down what you need to know and why, and it gives you a full rundown. Again, all the key terms you need to know are going to be highlighted red. So the question that most likely to come up is, why is it insoluble? Because there's no effect on osmosis. But you could get a 1 marker on what type of glucose is it used to make amylose. Could be just a simple 1 marker. It might also be compare the structure of, the most common is comparing the structure of cellulose and starch. And that's the question actually we're going to look at right now. Describe command words. So whenever we're looking at questions, we need to think of a few things. Before we start even thinking about the answer, teachers will often say, read the question carefully, read the question twice. I mean, this is good advice, but it doesn't tell the student exactly what you need to know. So first thing we're going to do is identify the command words. Second thing we're going to do is see how many marks it's worth. So it's a describe question, and then we've got 5 marks. Okay, so in my head, I now know the basic structure of what I need to do to get the answers here. I'm going to describe how the structure of starch and cellulose is related to their function. So the whole crux of this question is structure, linking the structure to the function. So if you don't, you could list cellulose is like this. Cellulose is like this. You can give me a list of structures, and then you give me a list of functions. But if you're not connecting this structure to that function, no marks. Even though it's clear that you kind of understand the stuff, the question does tell you, describe how the structure and cellulose are related to their function. So we must, in this question, link structure to function. And we've got 5 marks. In my head, I would make this a 6 marker, because then I can go starch for 3 marks, and then I can go cellulose. And I might not exactly get, but this to me is 2 3-mark questions strung together. It's not saying compare and contrast glucose with cellulose, starch and cellulose. It's saying link the structure of starch to its function for 3 marks, and it's saying link the structure of cellulose to its function for 3 marks. And then we've got the mark scheme here. Okay, so how much of this relates to the video that the screenshot of the video that I just put in there? Helical or spiral shape. So it's, in fact, what I might do, I'm just going to pinch this. Is it going to come up? Ooh. Okay. Let's see if we can pick out any of the answers here. Boom. Okay, so we've got helical or spiral, so it's compact. Marking point number 1. Okay, coiled, compact, 1 mark. We've got large molecule, insoluble, osmotically inactive. That's down here, 2, marking point 2. We've got branched. Okay, amylopectin is branched, so glucose is easily respiration. That's marking point 3. And large molecule, so it can leave the surface cell membrane. It's probably not in this. That's in the other video, transport across membrane. So we've got 1, 2, and 3 so far. Cellulose. See how they've subdivided? 3 marks, 3 marks. It's their choice. They didn't tell you to do that in the question, but when you analyze the question for 2 seconds, it's obvious that's what you've got to do. Long, straight chain, beta glucose. Boom, marking point 5. Joined by hydrogen bonds, marking point 6. To form microfibrils, marking point 7. And provides rigidity and strength. I'd probably say it, but I haven't read it. Either way, we're at 7 marking points in 1 diagram. That is the perfect answer to that question. So it's not rocket science, but you do need to know how to tackle the questions, because if you don't, then it's very tricky. Moving on, proteins. Okay, again, you should be able to, when you've revised this, you should be super tight. I'm not going to do the descriptions. It's going to take me too long to write, but you've got primary structure. What is the primary structure? If you can't give me a 1-sentence clean definition of the sequence of amino acids, again, we could draw it. We could have that being 1 amino acid, triangle. We could have a circle. We could have maybe a pentagon if I can draw one. Maybe we get 2 pentagons in a row because amino acids can repeat. Maybe we go back to a square. Okay, so the sequence of amino acids. What bonds are involved here? Most common. And again, it's not coincidence I've picked these things as the categories for these tables. These are really nice summary tables. You can use these in your revision. So peptide bonds. Secondary structure, we've got the alpha helices, the beta pleated sheets. They almost never ask questions on this. Almost never. I'm not saying it's not going to come up. It is in the spec. You do need to know it. They're held together by hydrogen bonds. The only thing that they are ever going to ask you about that in any great reality is what bonds are involved in the coiling of the alpha helices or the beta pleated sheets. Hydrogen bonds. Okay. Tertiary structure. What are the bonds in the tertiary structure called? Well, we've got the disulfide bridges, bonds or bridges. For bonus marks, does anyone tell me where does the disulfide bridges? They are covalent bonds. They take place between the cysteines that contain sulfur. That's really not all that needed to know, to be perfectly honest with you. Disulfide bridges, I didn't put an S on it. What are the other ones? We've got ionic interactions or ionic bonds. They're not too bothered about the second terminology, ionic bonds, ionic interactions. What else have we got? OCR are going to have a heavier element on the hydrophobic, hydrophilic interactions. AQA a little bit less so, but that's basically some regions of that tertiary structure are going to be hydrophobic, and they're going to fold on the inside. And some bits are going to be hydrophilic, and because they're in the cytoplasm, which is an aqueous medium, they're going to be on the outside. So that's basically same as the phospholipid bilayer. The hydrophilic bits on the outside, the hydrophobic bits on the inside. Quaternary, what do we mean by quaternary structure? The question that they always ask is, what does it mean? How do you tell this protein is quaternary? It's made from more than one polypeptide bond. More than one polypeptide chain. There's lots of peptide bonds there, all between all of the amino acids there. And the bonds is going to be all of the above, basically everything from all of them. Happy? Move on. Proteins, pretty straightforward. Yep. Okay. Let's go. How's the pace? Too fast, too slow? Good, good, good. Covering more content. Okay, so I'm trying to cover 12 months worth of content in an insignificant amount of time. Okay, let's make this just a touch wider. Okay, so here we go. Describe. This is like a classic, like describe everything. Tell me all you need to know about hemoglobin. So describe the structure of a hemoglobin molecule. This could effectively, you are required to know a little bit about hemoglobin, but this could be ATP synthase. Oh, my pen's not working. Yeah, this could be a protein, basically any protein. There's going to be a couple of these marks which will be specific to hemoglobin. But most of these 7 marks will be available for any protein. So let's start with those. Again, I'm going to pull this answer away. What does it want us to do? It wants us to describe the structure of a protein. And then we've got a little bit of, in the quaternary, we're going to have a little bit down here about the heme groups, yada, yada, yada. Okay, so the sequence or chain of amino acids joined by peptide bonds. Oh, look, it's the exact things that are in the table, which come up over and over again. The secondary structure, we can talk about the alpha helices, the beta pleated sheets. We get marks for them held together by what kind of bonds? Oh, they're hydrogen bonds. Okay, that's what they want to know every single time. It's not random that they just asking this and I've picked a question that happens to reinforce that. This is what they ask about proteins. That's how it happens. Okay, secondary structure. Secondary structure, we have, sorry, I don't know why that's kind of irrelevant. The tertiary structure, we've got the polypeptide chain folds up. It's the final 3D resting shape. If you're looking for my textbook definition, it's the final 3D resting shape, further coiling. We've got other bonding, disulfide, ionic, hydrogen, hydrophobic or hydrophilic. We've got hydrophilic R groups on the outside, hydrophobic bits on the inside. Quaternary, here's where we get into the specifics about hemoglobin. So it's got 4 polypeptide subunits, 2 alpha chains, 2 beta chains, but 1 heme group per polypeptide. So there's basically, we look a little bit like this. Let's maybe choose some colors. Thinking about alphas. Betas can be purple, and they kind of interlock a little bit, with our heme group being the blue bit in the middle. And then we have our sort of final 3D resting shape. Okay, prosthetic groups contain iron, yada, yada, yada. Prosthetic groups, this is an OCR term. If you're an AQA student or Edexcel student and you're worrying about that, don't worry, because you don't get tested on it. Okay, let's look at this. This is probably, in fact, what I'm going to do here. I'm just going to go cover the answer up before you get too keen on it. HIV attaches to a specific protein receptor on helper T cells. Okay, so immune system, that's where we're going with this question, right? A low percentage of people have a mutation on the CCR5 gene. Does it matter what the gene's called? No, as long as there's not more than one, it's just the gene. We don't need to know the name of it. A low percentage of people have a mutation on the CCR5 gene, which codes for this protein receptor. This mutation results in a non-functional protein receptor, okay, that doesn't work anymore, non-functional. Command word is explain. Okay, you've got to pick out the command word every time. You've got to know how many marks it is every time. So what does explain mean? Give reasons for. So we can, the only command word I'm ever going to change on an A-level biology paper is explain, and I'm going to replace explain with give reasons for. Give reasons for how this mutation can result in the production of a non-functional protein receptor. This is a variation of the most common A-level biology question ever, bar none, bar none. This is effectively the most likely thing that if I could give a prediction of one thing that's going to come up in some capacity over the 3 papers, well, the answers to this will be on your answer paper into some, the question will be wrapped up in a different writing, but it will be the same. Okay, so in fact, I'll show you my lesson to this, and I'll show you a 2017 paper. I wrote my lesson for this in 2015. I'll show you a 2017 paper, and I'll show you how my answer that I wrote is basically word for word exactly the answer off the mark scheme for the 2017 paper, even though I wrote my answer 2 years before, because I can tell, I just know when they're going to ask this type of question. I know what the words on the mark scheme are going to be. Super nice. Loads of Kezia, was that one of my regular students, just like banging it in, ding, 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 ding. Great answer. Okay, so I'm not going to dwell on the details here. We've got 30 minutes down, we've got 30 minutes to go, and we've still got heaps of content. Okay, so what are we looking for? A mutation. So we've got basically delta means change in my shorthand. If I'm doing past papers, I use loads of shorthand just to make them go faster. I want to do as many past papers as I possibly can. So this means delta. Change in DNA sequence. This causes a change in the primary structure, which causes a change in the tertiary structure. It's not an enzyme. The classic question here is it does not form an enzyme substrate complex anymore. But this isn't, we can't say that, which is almost the answer I wrote down, but we're not doing that because this is a receptor protein. But we can say that it's no longer complementary to the molecule that fits in it. Shall we have a look at the answer? Hey, Paula. Ricardo. Solo estoy Ricardo cuando hablo español. Okay, so change in DNA base sequence, okay? This is how I do my past papers, but we get marking point 1. Change in amino acid or primary structure, change in tertiary structure, and change in bonding. Boom, it's that easy. Okay, so then let's have a look at my lesson that covers this basically. This is all of it's the protein shape question or the enzyme question. This could be enzymes, this could be antibodies, it could be protein receptors as the example we've just looked at. So the tertiary structure of an active site is complementary to the shape of the substrate and forms an enzyme substrate complex. Or we could say an antigen antibody complex or antibody antigen complex. The question that we just looked at was this bit here. If there's a mutation, how does it cause a non-functional protein? Well, there's a change to the primary structure, change to the tertiary structure, change to the heteroactive site. The substrate is not complementary. There's no enzyme substrate complex. That is it. The only other question they're going to ask that in any other way is that if it's denatured, then we're going to break down those internal bonds, which changes the tertiary structure, which then feeds us back into this bit, changes the shape of the active site. Substrate is not complementary. There's no enzyme substrate complex or no antigen antibody complex. Okay. Transport across membrane is another massive topic. We're going to skip through this as fast as possible. These tables are going to be, as I said, like as quick as way we can summarize it as possible. They're all based off past paper questions. We're going to maybe do a few more past paper questions towards the end. Let's go down. I reckon that these ones. Okay, so diffusion. I'm just going to put P, osmosis, passive, facilitated diffusion. In fact, I'm going to do this in the corner. I'm going to do passive. Passive, there's 2 types or 2 molecules. Active transport, the clue is in the name. Okay, endocytosis, active or passive? Active and active. They do moving basically it's like the spindle fibers when you do mitosis, the cytoskeleton inside the cell to do that budding, to push out a vesicle or to envelop something from the outside. That moving those is quite energetically taxing, so it does require ATP. So that's often something that catches students out. They're not too sure because you don't cover it in very much detail. So both these are active, uses ATP. Okay, the 2 different types of molecule. Okay, what moves? I'm going to do what moves. What moves by facilitated diffusion? I'm going to hop about a little bit here. Okay, so we've got large molecules. Ooh. Come on, internet, you can do it. And we've got small, glitching out. My handwriting isn't that bad. My handwriting is pretty terrible. If you watch enough of my videos, you'll see. Small and charged, not. Okay, so what type of molecules, let's move backwards now, move by small charged molecules. What kind of protein? Go through polar would also be fine there as well. Okay, so this is a protein channel. So a channel is like small and skinny and thin and long, right? I don't know if you can, yeah. So that is, it's like narrow. So it's only going to fit something small through it. What kind of molecule does large stuff like glucose? Oh, that's not what it is. Also begins with C. It's a carrier. I think here aircraft carrier, which is a massive, great boat that has airplanes on it. If I could draw. Yeah. So an aircraft carrier, a massive boat, it carries massive molecules. Glucose, compared to water, glucose is massive. Yeah, everything's tiny for us, but water is 3 atoms. Yeah, H2O, and we've got glucose is many, many atoms. So these are large molecules. We've got water. Again, I'm going to use shorthand in my notes. Basically, direct diffusion. Okay, so what goes direct diffusion through a phospholipid bilayer, which is basically how they're going to ask the questions in the exam. Obviously, diffusion, if I fart here, it's going to diffuse through the air. It does not going through a membrane. It's just like permeating throughout the room. But if it's going through a phospholipid bilayer, it's got to be fat soluble, lipid soluble. Well, okay, active transport, it's going which direction? It's going to go against the concentration gradient. Okay, endocytosis, exocytosis, we're going to be moving vesicles in, vesicles out. I think we're done on this. Let's move on. There's so much to get through. Transport across membranes is a relatively basic topic. I'm sure you're probably okay. This question is going to basically answer that. Okay, so this question is not the direct tell me what you know about transport across membranes. Let's analyze the question. We've got describe as the command word. We've got 6 marks. Describe how proteins are arranged in the plasma membrane and the part they play in transporting substances in and out of the cell. Okay, this is a bit weird, how they are arranged in the plasma membrane. Okay, so, and the part that they play in transporting substances in and out of the cell. This is a little bit, think it through rather than just like describe how things move across the membrane, which is a question you do get sometimes. So how are they arranged? Well, we've got effectively 2 types. I actually only looked at this question the other day. That's our phospholipid bilayer. We've got some which sit partially in the membrane, and we've got some like transmembrane proteins that go all the way through the membrane. So in terms of arrangement, these were the only 2 I could think of when I did my live class, and I'd actually not seen this question in like 18 months. So that's transmembrane and partially in the membrane, or only in one layer of the phospholipid bilayer. And what role do they play? Well, we've got facilitated diffusion. We've got channels taking small charged molecules. Again, these are the facts they want you to give. We've got carrier proteins, which take large molecules. Always give an example. Small charged, e.g. Sodium, potassium, calcium, any of those things. Then large molecules, e.g. glucose, and then we've got active transport going against the concentration gradient. You kind of get the picture. You gonna let me delete it? Yeah. Some proteins pass right through the membrane. Some proteins are associated with one layer. Okay, that's the way they've chosen to put it here. It's involved in facilitated diffusion, involved in active transport. Protein acts as carriers. Carriers change shape or position. Proteins form channels or pores. Proteins allow passage of water-soluble molecules. Okay, they're called aquaporins. There's really very few references to aquaporins in the spec, so I don't tend to go into too much detail on them. This specific paper this came from, can I tell from their answers? Normally I tell by the font, but I've changed the font, so it was either OCR. I don't know. It's relevant to all of you. And again, this is an OCR question for sure. Some molecules move across the cell surface membrane by passing down a concentration gradient through the phospholipid bilayer. The movement of some polar molecules across the membrane involves carrier and channel proteins. When this movement occurs down the concentration, it is called facilitated diffusion. And when it goes against concentration, it is called active transport. These are kind of more straightforward kind of questions, but they're really good for summary. Like you're going to have, if you can't do these like snap, 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 then you need to go back and tighten up your knowledge, because when you get to those application questions, if you're trying to fiddle in the details and like slot it all together in the back of your head, oh, how does this work? And then you're trying to figure out how the system works on top of the minor details, it's going to be really difficult. So the application becomes easier when your facts are nailed down. And these are the facts you have to know. Otherwise, you're going to be really struggling. Energy in the form of ATP. Okay, let's have a look at this. I've kind of lumped DNA and protein synthesis here. because you can see the similarities. I'll put the exam order. This question on the left was an AQA, although I didn't actually write that down. Okay, so describe how a nucleotide base sequence in a gene is used to synthesize a polypeptide. Okay, that basically, what's that saying? Let's rephrase that question. I'm just going to cover it up for just a second. I'm not too worried about the answer. What's that question really asking us to do? Let's rephrase that question in a way that makes it a bit easier to answer. Describe how a nucleotide base sequence in a gene is used to synthesize a polypeptide. Simplify that as a question. Talk about, okay, so talk about, I would say, fine. What's the command word? The command word can't change. We kind of have to keep the command word. The only command word I'll tweak is explain and replace it for give reasons for. Okay, sweet. So describe, I'm going to put this in 2 words. If I could write, which I can't right now. Describe. No, in fact, 3 words, because it's both. Protein synthesis, or the process of. Describe how a nucleotide base sequence in a gene is used to synthesize a polypeptide. Describe the process of protein synthesis. Edexcel, describe how the sequence of bases in a DNA molecule will be used to form the primary structure of a protein. Same question, different question. I need some water. Same question, different exam board, different wording, same question. We'll do these 2 first. The next question is on DNA synthesis. Okay. Let's have a look and see. Transcription, the DNA is copied or transcribed into messenger RNA. Free RNA nucleotides are added. I'm going to just pick out the bits that I think are useful here. So yeah, transcribed. Yeah, it's kind of all relevant. They line up by complementary base pairing or described. So that's A with T and C with G, and G and C. Doesn't matter which way around you write them. One template or reference to the sense strand. So one of the differences here in DNA replication, we're splitting the anti-parallel strands, and both strands act as the template. In protein synthesis, only one of the strands, the sense strand, if you want to call it that, it's not that important, doesn't come up too often in terms of that key term. Only one of the strands is going to code for the messenger RNA. Catalyzed by the enzyme RNA polymerase. Always give the enzymes. If an enzyme is involved that you need to know, so the enzymes are that you need to know RNA polymerase, DNA ligase. If you're doing A2 biology, we need to know ATP synthase. We need to know about, what else pops to the top of my head? DNA helicase, yeah, some of those. And then translation, RNA moves to the ribosomes. Transfer RNA molecules bind to the messenger RNA. So the anticodons pair with the codons. So key terminology here is that we need to say that DNA has triplets. Always made mistakes. DNA is triplets. Messenger RNA is codons. Transfer RNA is anticodons. Those 3 are always badly used in exam papers. People talk about DNA codons. There's no codons in DNA. DNA has triplets. Messenger RNA has codons. Transfer RNA has anticodons. tRNA codons pair with complementary codons. Each has a specific amino acid attached to the transfer RNA and the formation of peptide bonds. They always want to know about the bonding. Always want to know about the bonding. This is basically the same. You can read it yourself if you need to watch the recording. Okay, let's see how this correlates to what's the similarities between this and DNA replication. Okay, so I'm going to change color. So this, for some reason, they always want to know about helicase when you're talking about DNA replication. They're not interested in it when you're talking about transcription and translation over here. But same enzyme does the same thing. How do you think the strands separate when you do, whoa there? It's not what I wanted to do. Let's go, not 3%, maybe. Oh no, had a bit of a meltdown. We've got spinning wheel of death. Okay, so whilst we've got no screen, you can bullet point me the stages of DNA synthesis, DNA semi-conservative replication. You just saw the mark scheme very briefly. Whoa, everything's happening right now. Okay, let's go. Where are we? We're somewhere over here. I have a feeling you might. In fact, I'm going to leave it small. Let's do this. Let's test ourselves. Okay. And these are kind of things like really good, simple tests that you can give yourself. Okay, so off the top of my head, without reading it off the mark scheme, DNA helicase unzips the DNA, breaks the hydrogen bonds. Yeah, both strands act as the template, not just one. Both strands act as the template. Yeah, new DNA nucleotides join on according to complementary base pairing, A with T, C with G, joined by DNA polymerase, the enzyme that polymerase. So A's is the enzyme, polymer is the enzyme that makes the polymer of DNA. We're making new DNA, therefore it's DNA polymerase. What else can we say? We talked about complementary base pairing. This forms phosphodiester bonds, another key term. It's sandwiching as many key terms as you can. If it's a relevant key term here, then you have to put it in there. We're going over here. Is where we're going. If I do this, we kind of do. Ooh. Okay, how good did I get? Complementary base pairing, DNA polymerase, forming phosphodiester bonds, hydrogen bonds reform, semi-conservative DNA replication, a key term that I didn't include. Okay, so semi-conservative DNA replication, because, and we can say that DNA molecule contains one new strand and one old strand. That's what they like you to say. If the question is specifically on semi-conservative, then it will say that's how it needs to be. Essentially, that you got one new strand, one old strand. That's why it's semi-conservative. So half of it is retained. Okay, how do the lessons compare to this? Let's have a quick look at that. I wonder if we can get answers on the page at the same time. Almost. Okay, so DNA helicase separates the hydrogen bonds. 3 nucleotides attached to the template strand, complementary base pairing, RNA polymerase join nucleotides together. In fact, let's zoom in a little bit. I think that's probably too small. Again, these lessons are all written before the questions were written. It wasn't lucky that I guessed the right bits. It's the bits that come up all the time. Hydrogen bonds reform. And there's a little bit on the differences between DNA, prokaryotes and eukaryotes and how they're different. I'm not going to go into splicing right now. That's another topic for another day. Okay, and DNA replication, how is it? DNA helicase unwinds the DNA, breaks the hydrogen bonds, complementary base pairing, A with T, G with C. New nucleotides pair, both and acts as the template. DNA polymerase joins by condensation reaction. And then there's a little bit there, which I think is going to come up about the active site of DNA polymerase being complementary to only bases in one direction. So if we think about our nucleotides, we have our deoxyribose as our sugar. It's a pentagon, right? It's a pentose sugar. What do I mean by that? Well, let me just sketch this. So if we've got our pentoses of our nucleotide, so there's our pentose, we've got our base here. I'm going to just do A for just and this goes up here, and we've got our phosphate group here. So let's say we've got our DNA polymerase. The active site of DNA polymerase is going to be complementary to the nucleotide in only one direction. It can only join another nucleotide onto this in this position. That's it's the way that let's say this one's a T, doesn't really matter which one it is. It cannot, so in order to write, it can go continuously in one direction, but to do it in the other direction, it can't go down. So what it has to do is do a little bit up, and then it gets joined together, a little bit up, and it gets joined together. So this is why, if you've heard of our sucky fragments, that's what they are. But I think having the active site of DNA polymerase, or RNA polymerase for that matter, being complementary to the nucleotide in only one direction is something that I have a feeling they're going to investigate and test you on at some point during the next set of exams. Okay, let's look at a practical application question. Oh my God, it's 10-2. This is a bit more of a translation question. I think we've covered this enough, but basically it's saying protein synthesis is stopped by tetracycline. Explain why. Give 3 reasons why this molecule, and at this point I'd sub out tetracycline. I wouldn't even bother with tetracycline. Something that does this, how does it stop protein synthesis? Well, they've deliberately not chosen the word transcription or translation in there. So translation is going to be the mark. If you ever notice the fact that they've gone out their way, here, protein synthesis. Which bit of protein synthesis is this going to stop? Will it stop transcription? No, we've already made the messenger RNA. It can't stop the transcription. It's done already. So it's got to stop the translation. It's going to bind to the codon. It's going to prevent the anticodon from attaching. And therefore, it's going to prevent amino acids being added to the chain. Therefore, translation is prevented. Let's move aside. This is, again, super classic question. The pancreas is an organ that secretes protease enzymes. Nice to know. Outline is our command word here, which is a bit of a weird one. These are all new spec questions. How organelles in the pancreatic cells work together to produce and release these protein molecules from the cells. So how the organelles in the pancreatic cells work together. Let's rephrase this question. Before we answer it, how would you rephrase this question? Outline. Why is the command word here outline? If this question was describe the process by which proteins are synthesized and exported, we've got to go transcription, translation, and we then got to go to the Golgi and package it up, and then exocytosis. That is a 12 marker right there. And it's just not, they're not going to ever give you a 12-mark question. The AQA students have to do an essay at the end of it to do 25 marker, but this isn't an essay question. This is just a 5 marker. That's why the command word here is outline. It's not wanting you to go into all the minor details. This is looking for an overview, organelle to organelle. Probably the organelle, what does it do? 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 That's the structure for this answer because the command word is outline. So we're going to maybe say DNA contains the genes that codes for the protein. We can then say the ribosome on the rough endoplasmic reticulum is going to do translation. When we're going to say a vesicle is going to transport the packaged up protein to the Golgi apparatus, which will modify the protein. It might add a carbohydrate chain, might add something else to it, might add a lipid to make it a glycolipid. And then it's going to package up in another vesicle, a Golgi vesicle, and transport it to the cell surface membrane because this is, it was saying about it being secreted. So it's got to come out of the cell. So the Golgi apparatus will package it up and then go to the vesicle where it will move out by probably exocytosis. Bueno. In fact, let's cover this up. Let's have a look at this. Again, super mega classic common question. The structure of a colorobacterium is different from the structure of an epithelial cell from the small intestine. Describe how the structure of a colorobacterium is different for 3 marks. Okay, so how is it different? What's this question saying? We can rephrase this. Describe how the structure of a prokaryote is different from a eukaryote, is what this is saying. 3 marks. Okay, booming answers going. What are the main differences? The DNA. We've got no, eukaryote means truly nucleated. It has a nucleus. Prokaryotes have a circular chromosome, a loop of DNA. You can say basically whatever you want in terms of that there. So in the DNA. So that's one major difference, the type of the DNA. You could talk about eukaryotes having histones and having DNA associated with histones. Fine, probably not a wrong answer, to be honest. That's perfectly acceptable. It's in the new spec, a little bit more is in the old spec, because now you look at acetylation and methylation of histones and how that can affect gene expression, but that's another story for another day. That's A2 content as well. Okay, so we can talk about membrane-bound organelles, but don't just stop at membrane organelles. They're always going to give you 2 chances of bites of that cherry. So mitochondria, Golgi, endoplasmic reticulum, any of those things we can say as a membrane-bound organelles, give 2 examples, always 2 examples. Then can we think of anything that's in a prokaryote that's not in a eukaryote? So we can say prokaryotes are missing mitochondria, they're missing chloroplasts, they're missing all these things. But what does a prokaryote have that eukaryotes don't? Talk about plasmids, Pili, if you know what a pilus is, but that's not too worried about. Talk about flagella. There's a bit of a grey area. People call, so technically sperm don't have a flagella. It's actually something slightly different. I got questions on whether flagella sperm have flagella or not. But we can say, you can legitimately say that prokaryotes have flagella and eukaryotes don't, because technically the eukaryote one's a slight, yeah, you got it, you nailed it. Who was that? I can't even see. The Undulapodia. You got it. That's exactly what it is. I can never remember because I never teach it. Okay, and then we've got the answers there. Capsule, flagellum, plasmid, cell wall, etc. You could talk about the structure of the cell wall. Possibly you could say it's peptidoglycan versus cellulose if you're comparing it to a plant. No, we're not comparing it to a plant. We're comparing it to an epithelial cell. So we don't have to worry about it being, we can't talk about plant stuff there. We can't talk about chloroplasts. Peptidoglycan murin, you may have called peptidoglycan murin. It's the compound from which prokaryotes make their cell surface membrane. Okay, again, huge topic, and this could come up in a million different outfits. This question, it could come up in a zillion different ways. But again, this is probably second most common questions on the A-level biology exam. Again, they're not going to just ask you flat out which 3 factors affect the rate of diffusion. Temperature does. The only place in which temperature will come up in terms of the rate of diffusion will be in nerve transmission, because in warmer organisms or warmer climates, the diffusion of sodium and potassium across the axon membrane will speed up the rate of nerve transmission, but that's the only time. Otherwise, we in humans are homeotherms. We are thermostatically controlled by homeostasis, and therefore when temperature is not going to fluctuate, that's why they don't ask. Okay, in no particular order, surface area. What about another one? Okay, Edexcel students are the only people who know about Fick's law. I'm not going to talk about Fick's law of diffusion today because that's not covered by OCR and Edexcel. We can talk about the concentration. This is my shorthand for concentration gradient. Closer to things are, the easier it is going to be for diffusion. Capillary endothelia being 1 cell thick, and also the alveoli being 1 cell thick. The closer they are together, the shorter the diffusion distance, the greater the rate of diffusion. So that could come up in millions of different ways. Okay, I need to stop relatively soon, but I'm gonna crack on and get as much out of there as possible. Okay, so basic structure of the heart, always drawn from the front. You're never gonna get a diagram drawn from the perspective of the person who owns the heart. We should be able to label A, B, C, D pretty easy. Again, this is like spot knowledge that we kind of need to know. So we've got the aorta, we've got the vena cava. Okay, we've got the pulmonary. Pulmonary means lung, so going to the lungs, it's going away from the heart. This is the artery. Arteries go away from the heart. Forget about oxygenated and deoxygenated for arteries and veins, because these are exceptions. And then we've got the pulmonary vein, because it's returning. It's going away from the lungs, towards the heart. Okay, so what's X and what's Y? Here's the AVN, if my graphics tablet will write for me. Y is, okay, this is the bundle of His moving into the Purkinje fibers down here, is what they are. Yeah, septum is the central part. I'd say because it's coming directly out the AVN, I'd say this is more the bundle of His than it is the septum, which is the divide between the two halves of the heart. Okay, what does the diagram suggest about the pressure in the atria compared to the pressure in the ventricles at this stage of the cardiac cycle? Explain your answer. So 3 marks to give reasons for. One is we're going to get 1 mark for stating the pressure in the atria compared. Any type of comparison, we must use big small great less If you don't give me a comparative in that sentence, you will not get a mark. You must clearly demonstrate one being bigger than the other. Effectively, that's how it's going to work. Okay, so if the atrioventricular valves are open, that means we must have high pressure here and low pressure here. That's how it works. Otherwise, if it's the other way around, these are one-way valves. They'll shut going out the other way. You're not going to be able to get any blood flow going back through the heart. It's a one-way system. Explain your answer. The atrioventricular valves are open, and therefore blood is flowing from the atria to ventricles, and to prevent the backflow of blood, they're one-way valves. Can I delete that? Yeah. High again, we must be using those comparatives. I'm surprised actually that these aren't in bold or underline. Atrioventricular valves open. Where's the 3rd mark? Credit score. If I must have miscopied that. There's a 3rd marking point there. It's not on the page. Cycles. Mathematical element, 10% of your papers. Every paper will be mathematicals level 2 higher GCSE. Okay, so at these different times, this is volume of blood in the right ventricle, time. The atrioventricular valves open. So at 0.2, I've highlighted it here. This won't be done for you on the exam, but I just pulled it out to make it a bit more straightforward. How do we know if the atrioventricular valves are open or closed? So this is the volume of blood in the ventricle. So volume of blood is going down, it's decreasing. This means the ventricle is emptying. So if the ventricles are contracting ventricular systole, then what must the atrioventricular valves be doing? They must be closed. Because blood, if we draw our sketch of the heart again, we've obviously got some blood vessels coming out here. Okay, so right now, the volume of the right blood is coming out here, which means the ventricles are contracting, which means the atrioventricular valves must be closed. Everyone with me on that? And if the blood's leaving out the aorta, then the semilunars, oh no, I'm doing them the wrong way around. Atrioventricular valves, the semilunar valves are open. At 0.6 seconds, what's happening to the volume of blood? It's increasing. So the ventricles are filling at this point. If the ventricles are filling, then the atrioventricular valves must be open. That's not how you spell open. That's how you spell open. And if the atrioventricular valves are open, if these are filling, then what's likely to be happening to the other guys? Well, you've just pumped all the blood out. So the blood's here. Gravity wants to bring it back down. Also, you've got that elastic recoil stretch. When the ventricles contract ventricular systole, the aorta, the biggest artery in the body, is going to stretch. That elasticity is going to stretch right out, and it wants to recoil, which maintains high blood pressure and even flow of blood. But that would mean, if it could, water would flow back down into the ventricle immediately as soon as you get into ventricular diastole, as soon as the ventricle relaxes. But it can't come back down because the semilunar valves are closed. Okay, how many beats per minute is it? Well, how do we work this out? Well, we find something on here that's the same. Well, the only time we've got the same here is 125. So that means that one complete cycle takes 0.7 seconds. When you get these questions, it's always worth thinking, okay, I've got one cycle. How long does one cycle take? That's your first thing to figure out. One cycle. Okay, one cycle takes 0.7 seconds. If it's 0.7 seconds for one cycle, is it going to take, if it was 1 beat per minute, what am I going to get? I'm going to get 160 beats per minute. So if it's just under 1 second, I'm going to get slightly more than 60 beats per minute. So I'm thinking a little mental calculation before I begin this. I'm not going to whack in the calculator. I'm expecting a number a bit more than 60. Yeah, because 1 cycle is just less than 1 second. That means I'm going to be getting something. I'm expecting 70 to 80, something in that region. So if I do my numbers wrong on the calculator and I get a number that's less than 60, then I'm probably not right. So 60 seconds in a minute. How long does 1 cycle take? 0.7 seconds. 60. Can anyone do this? 80, 85. That I've actually got my scientific calculator here. Let's punch it in just to make sure that you're right. Divided by 0.7. Okay, so the number we get on the calculator here, 85.714. Oh, 28. Yada, yada, yada. Okay, so what's the answer on the exam? Am I giving it to 4 decimal places? Am I giving it to 10 decimal places? What are we calculating? BPM. I'll check the masking. I would give this to the nearest whole number in this case. So this would be 86 beats per minute. Half a beat per minute? Unlikely to be on half a beat per minute. Okay. Let's say I'm going to do another 5 slow minutes. In fact, let's skip mass transport. Let's skip this bit. Okay, so this is a great, again, summary diagram. What do you know about arteries or arterioles? Arterioles just small arteries, veins and venules. Venules just small veins. So lumen. So this lumen of an artery is actually smaller than that of a vein. So this is something that often catches small people out, smaller, because we've got this folded, wrinkly epithelium which can stretch. And then we've got a layer of muscle. That's our artery. A vein is basically big and hollow with little muscle, no folded endothelium. So the diameter of the lumen of the vein is actually larger. Okay, valves. Obviously present over here. Absent from muscle. We have more muscle, more muscular arteries and arterioles. Sphincters are just basically the things that can divert the flow of blood from one place to another. They're a tiny bit of the spec. People got really hung up on sphincters. Sphincters basically, we all know have a sphincter on our bum hole, but that's just an example of a sphincter. They have sphincters everywhere. They can basically divert the flow of blood. If we've just eaten a meal, we want more blood at our intestines. So the arteries taking oxygenated blood are going to dilate the ones going to our intestines, and they're going to maybe vasoconstriction to the ones doing our muscles, for example. Just fill that in. You're going to have loads of time to figure that out afterwards. Okay, again, I think I'm going to cover this up. I was just going to rip through them, but I think let's test you a little bit first. It's going to be better for you in your own good active learning. Okay, describe the processes involved in the transport of sugars in plant stems. Okay, so what's this question saying? Sugars in the plant stems. So what method or how are sugars moved, and what key term have they not used in the question? Translocation. Are we going to get a mark for translocation? You bet we are. 5 marks. Describe translocation. 5 marks. Okay, so we wanted to be talking about companion cell. You can say assimilates, you can say sucrose. I've seen questions on why sucrose transported. I'm going to do that in a second. Okay, so we're going to take sucrose. Companion cell is going to actively transport sucrose into the sieve tube element. It's going to decrease the water potential. So then water is going to move from the xylem and from the companion cells. All the surrounding tissues are going to go into the sieve tube cell by osmosis. And that's going to basically create high hydrostatic pressure. You've got all this water coming in. It needs to go somewhere. It's going to push out in all directions. And where is it going to go? It's going to push it from the source, which is where the sucrose or the assimilates are made, and it's going to go to the sink, which is either where it's going to be respired and used, or it's going to go to like the tubers. If it's a potato plant you're going to convert the sucrose into starch so you can store it for a rainy day, basically. at source, actively transported into the sieve tube cell. Companion cell, or you can call them companion cell is the right word to use here, decreases the water potential. Water moves in by osmosis, produces high hydrostatic pressure, mass flow or transport towards the roots. I would call it the sink if we're talking about translocation. At the sink, the roots or sugars are removed or unloaded. What's going on here? I'm moving. Let's not do that. Oh, let's actually take that away. Again, just reminding ourselves, how does the lesson prepare us from this? Well, in the source, high concentration of the solute, e.g. sucrose, active transport in from the companion cell to the sieve tube cell, decreases the water potential, water moves in by osmosis, creates high hydrostatic pressure at the sink. It breaks down or uses the sucrose, converts it from sucrose into starch, which increases the water potential. Water moves out by osmosis, and it basically, that's the end of the story. Okay. Let's go. Okay, so let's just remind ourselves of some of the competitions. I might show you some of those lessons inside the course too. So again, the live web class is coming up this week. You've got 6 o'clock maths, 6 o'clock chemistry today, 8 p.m. maths today. So I will make sure I finish on time so you guys can have a half a chance. Competitions. Let's maybe refresh this page and see if we've posted anything in the last hour. Okay, this is the post. Past paper training week, join now. The competition for the winning of Instagram, you have to follow the Instagram page and you have to tag a friend in this post. It's pretty clear how to find it. You've got to go to the comments, tag a friend. We will pick you and your friend, the person that you tag, will win a course of your choice for free if you're in the web class on Sunday. Sunday evening, 6 p.m. It's going to be a Q&A with me, with Ronnie, and with Richard, the chemist. You're going to be able to drop questions on us. We're going to throw you some exam questions, maybe give you some teasers. And yeah, it's going to be super duper useful. Okay, so that's how you can enter that competition. Follow the page and tag a friend. You've got to do both. If you don't do both, you don't get competition. Okay, Facebook reviews. Okay, let's close down some of these things, or people messaging me. I'm not using Facebook right now. Okay, so again, leave a Facebook review. We'll pick the winner at random. It doesn't matter how you review us, but you have to leave a public review so we can see it, and it has to have some writing in it. You can't just leave a star rating. You have to basically leave some writing. We will pick a winner on Sunday, and you will win the course of your choice absolutely free. Okay, who wants to know about an unlimited pass? Who wants an unlimited access basically to the course for the weekend? I'm going to throw this out there as a poll. All right, it'd be maths, chemistry, and biology, all subjects. How many of you voted? 60%. Let's get that over 80%. You want to know how to do it? Okay, let's publish that. Okay, 98 too. I'll take that. Okay, I'll show you now how to do this. Now, in order to do this, basically all you've got to do is bring a friend. It shouldn't be too difficult. Any A-level maths, any A-level chemistry, any A-level biology friends. So you'll find this page on our website, taylortutors.co.uk. It's unlimited-free-trial. You're not going to find this in the menu. This is for you guys only. Unlimited-free-trial. And you guys are going to be able to, basically all you have to do, as I said, to bring a friend is, scroll down. There's a form here, your email address. We will send a specific coupon straight to you, to the email address you give us here. We just need your name and how many you want. You've got to bring a friend, so the minimum you can do is 2, 1 for you, 1 for one friend. If you just want to bring 1 friend, that's absolutely fine. You can have a maximum of 10, so you can bring 9 friends with you, basically. So you can share the love with your classmates, with your friends at other schools. Basically, you're just going to have to send them the link that we send you and the coupon that we send you. And that's all you're going to have to do to join the course. Okay, I think we're kind of good on that. What are we at? We can do questions for a few minutes. Let's pull up. I might just stop screen sharing now. Okay, well, I mean, within the course, I had loads of questions, in fact, from students who were just asking me about seeing inside the course. Those people who haven't, like over a third of you, I think, hadn't seen the course at all. This is right now, I happen to be in the AQA AS biology course. So this is how the course looks once you're inside it for all the paid members. And when you get your free unlimited access pass this weekend, you'll be able to dig around and do all the kind of things. We've got in here a copy of the specification, which you can download. And this is basically, again, this is the AQA one. There's one for OCR, there's one for Edexcel. And within here, it's all the spec laid out with links to each of the videos. So let's say you want to go through and you want to study, I don't know, let's pick a topic. Oh, I think let's just pick one that's going to load. Okay, the Burette test for proteins. We could then click on this link and it will take us to the right lessons. You can download this, you can go through this. But if you know that there's areas of the spec that you're weak on, you can click on that and you can jump through. It will take you to the videos that you need. Super duper useful. Okay, what else can we go through within here? So it's all laid out in the same order as the specification. The OCR course will mimic the OCR spec. The Edexcel course will mimic the Edexcel spec. So you can pick your topic. If you're struggling to find a video, you can do like Command F and you can just do a little search. So if we search for, I don't know, DNA, if I was typing in there, it would help. Yeah, and then we can find any of the lessons that are on DNA, but we can also use the specification. So it's super easy to find courses in here. There's quizzes, so you can answer questions, all that kind of stuff. Okay, sweet. I'm going to pull open, I'm going to turn off my screen sharing, and I'm going to open my Q&A panel, and there's a heap of questions in there already. I don't want to get rid of that. Interesting. I want to view my Q&A panel. Okay, sweet. You guys can just see me now, right? You can tell me answers in the chat. Awesome. Full screen. Hello, everyone. We're on YouTube too. We're on YouTube Live. Anyone watching on YouTube, you can't leave comments. Sadly, you've got to join the live web classes from academy.tailoredtutors.co.uk or go to the Tailored Tutors website. There's free resources in the menu. Find the course through the free resources in the menu. Okay, right. You can stop asking your questions right now because currently they're ripping off my screen faster than I can actually read them. So just hold on. I'm going to go through all of these as much as I can. Okay, so where will the recordings be? Where you click to join this live web class, that we're going to post the recording there. Let me make this a bit bigger so I can have half a chance of reading them. Why are biology exam questions so stupid? They basically are, whoa. Yeah, I said stop leaving me questions because I can't type them fast enough. Okay, you won't be emailed a link. No, the link to the recordings will be put where the sign up button was. Is practical skills not included? That you can, the practical skills course is all included with the revision kit. So if you get the revision kit, if you want any of these resources, then the revision kits are available. So there's biology revision kit, there's the maths revision kit, and the chemistry revision kit. That's the only choice. You can buy that or the live web classes. The live web classes that we're doing between now and the exams, we're doing a full series of like more information delivery web classes. We're going to be doing them for biology, chemistry, and maths. You can buy either the web classes or you can buy the kit or you can buy both of them. There is going to be a bit of a sneaky deal. If you attend those web classes on Saturday and Sunday, there's going to be a little bit of a promo that you guys can make the most of as well. So do make sure you stay tuned for those web classes on Saturday and Sunday. Okay, can you watch it later? Yes, you can. What is a transmembrane protein? It's a protein which crosses the span of both, goes from outside the cell surface membrane to inside the surface membrane, completely through the phospholipid bilayer. I can't cope with the questions that you're asking me right now. So DNA ligase, oh, it's gone off the screen. In fact, I'm going to just go to the bottom. Screw that. It's going to be easier. Otherwise, I'm trying to... Yes, there's videos. All the full Edexcel spec, the full OCR spec, and the full AQA spec are all covered. Suggest questions. Okay, suggest questions are going to take something that a principle that you know, and there's a full, and you're basically then going to have to take that element and don't go too deep into detail of suggest questions. It's looking for a sensible suggestion, and therefore, please don't repost questions because at the moment they're just ripping off the screen. I literally can't read them most of the time before they've gone off the screen. So just hold on with your questions. Otherwise, I can't answer any of them. So suggest questions, take your, take the things that you know. So if it's about a protein. Yeah, maybe it's a protein you've never heard of before. It's doing a weird thing. It's something you've never actually sort of come across in any capacity. Well, that's not too bad. It's okay. Don't worry about that because it's asking for it to be a protein. So, okay, put yourself in the mindset of like proteins. It's going to have peptide bonds. It's going to denature if you heat it up too much. The bonding inside is going to get all weird if you expose it to high pH or low pH, all those kind of things. Evaluate questions. Hannah asked me. Evaluate questions, you've got to give the pros and the cons. You must give both sides of the argument. If you don't give the like, okay, why was the experiment good? Okay, the experiment used a large sample size. They calculated a mean. They did a statistical test. However, they only tested female mice. They didn't test male mice, or they only tested mice. They didn't test it on humans. Yeah, so these kind of things. What is a chromatid? A chromatid is when we've got, I'd have to draw that. I'll jump into the web class in a minute. Okay, so the AQA essay question, there's a full web class going on the AQA essay question. I'm not going to delve into details now. There's also some lessons within the course that I'm going to publish on the AQA essay question. 3 weeks to the exams. No, have you left it too late? Definitely not. Use the course. The course is the most streamlined way to learn the content. We've had students complete the course in a week or so. So it's definitely not too late. Even after this weekend, you can definitely push up, I'd say 2 grades using past papers and using the courses that we've produced. You can easily increase 2 grades between now and then. Okay, what else? What question have you got? I can't read them. What are the critical values for t-tests or any statistical tests? You're going to produce a critical value. If the calculated test statistic is greater than the critical value, then you reject the null hypothesis, accept the hypothesis that there is a statistically significant difference between A and B. Best way to revise A-level biology, use the course, do past papers, revise. What does the A-level biology test you on? It tests you on how good you are at the A-level biology exam. So do loads and loads and loads of past papers, use the videos as much as you possibly can. If you need to take notes because you're relearning stuff from last year, then obviously that's going to be the most effective way to use the course. I would say pinpoint the areas you're weak on, go back and write notes, and then target the bits that you definitely need to brush up on, and then use past papers to fine tune your exam technique for the bits that you do know. So using the course, the videos that we've produced, plus the exam technique is by doing past papers is the best. So can you pay for basically the course is the biology course covers the entire spec. All of the course is covered in the biology spec, which is why it's slightly more expensive. The other 2 courses are revision courses, which are going to be super honed on helping you get better grades in the exam. So the biology course is £119, including VAT, and the other course are £89, including VAT. Why is a polysaccharide not a sugar? It's a polymer of sugars. It's a carbohydrate, but it's not is it a sugar? It's made, no, I'd say it's a carbohydrate, but it's not sugar. Okay, what was the question? You're doing AS exams. Can you get free access? Definitely not. No, I spent tens and tens and tens of thousands of pounds. I've paid teachers lots and lots of money to create these courses for you. Unfortunately, like these awesome videos can't continue unless people pay for them. So there's the way to get free access is to enter the competitions and to be the most engaged and most interactive person on here. You can win them. Will I be doing for the A2 exams as well? I haven't quite decided. I can't remember what I'd probably be doing this. Did you find today useful, first of all? Did you find that ACE? Did you find today? Yeah, you did. Okay, so, okay, I'm going to be doing, yeah, okay, well, I can do the next web class as being an A2 recap. I'm going to do an exam technique, a little bit thrown into that as well. How can you access the A2 cheat sheet? Attend the web classes on the weekends. Attend the web classes on the weekend, and I will make sure that you can get the cheat sheet somehow. Yeah, okay, absolutely. Who asked me? So who's used the course and who wouldn't mind shouting out to some of the people who've used it, give you an impartial, let me know in the chat, because if you leave it in the Q&A, you just come up as anonymous. If you leave it to me, if anyone wants to go through and just shout out to other people to how their experience of using the course this year has been, then I can just turn your mic on and you can have a quick 30-second chat with me and tell people what you thought about it. You will need a microphone or something on your device. I won't turn your video on, but I can turn your mic on. If you've got a pair of headphones and a mic on, that might also work. I can't remember who it was. There's so many people in here. I'm trying to, the best thing ever. Found it very useful. Okay, ultimate guide users, anyone who's been using the course, what are your honest reviews of the course? You can chuck that in the chat to all the people who are viewing this, so they can, it's not coming from my mouth then. I know how effective it's going to be. I know how much it's going to improve your grade, but get it from the words from the people who've been using it all year. Okay, so can you use bullet points in the exam? Yes, unless it's an extended essay question, something like that. Yes, you should use bullet points 100% in the exam. I need to open the chat so I can see it. We've got a few minutes left. How do I pop open the chat so I can, I want to. Best thing ever. Why can't I see? Selena. Okay, Selena, I wonder if I can open you up and to go, who can I view? Panelists, attendees. I'll see if I can find you, Selena. Selena, do you have a mic on that you can use? I can maybe lower hand. I can promote you to a panelist. Let's try that. Although, let's give this a go. I'm working. I want that off the screen. Okay, you're going to rejoin as a panelist. I now need to view panelists. And okay, your video is off. Selena, can you hear me, or can we hear you is the question.
Hi, everyone can hear me.
Yeah, wonderful. It worked. New technology. I'm figuring it out. It seems like you can hear me. Okay, so I would say, when did you start using the video, Selena? I don't have any idea, so you can tell me.
So basically, I got this course at the end of last year because I got an E in my mock.
Okay. Okay, so you've been using basically all of your A2 year, all of year 13?
Yeah.
And okay, general, what do you think about it in a few words?
So basically, I think it's one of the best biology courses out there because all the videos are already condensed and it just gets straight to the point. And in my mock exam this year, I got a high B. So I've gone from my E to a high B.
Awesome. Well, and it's so nice to hear that. Obviously, from my perspective, I wrote the course. I know I was basically tutoring students and doing that kind of exam performance with them, taking them from E's and D's to A's and B's. And it's nice to know that the course is doing that too. Does it save you? How does it compare to a textbook, Selena?
Well, it's way better than a textbook because I barely use my textbooks, to be honest. I just watch the videos because it just gets straight to the point and all the information is there and it's really well explained.
Sweet. And do you feel much more confident going into your exams now that you've been using the course and like summarizing it?
Yeah, I think I'm ready for it now.
Ready for it. That's nice to hear. And in terms of like how quickly you could cover the course, like with a month to go, do you think there'd still be value in ripping through the course and learning the courses as quickly as you can?
Yeah, because I went through my AS course quite fast, and basically with the A2 videos, I was watching them after I finished every topic. So I was like consolidating my revision through the courses, like through the video.
And you think a month would be enough to get like good value and improve a grade or so?
Yeah, I think you just have to keep consistency and just like keep watching them and just putting the time into it and do it.
Do you feel like the way that I've laid it out and using the mark scheme answers in the videos, do you feel that helps you get the right words in the exam? Like when because one of the main complaints is that you read a question, you write a pretty good answer, but you haven't quite hit, they're so specific, those marking points. Do you feel like the course helps you use the right words in the exam to get the marks?
Yeah, because the notes from the course is basically mark points. So I think that's really useful because it just gets down to the point and it's like, there's no waffling and stuff like that.
Well, that's exactly how I designed it, and it's super nice to hear that it's doing really well. Anything else? What would be your advice to those students out there who are thinking about buying it?
Go for it. I think you should get it because it's worked really well for me and really well for other people. So I think you'd at least go up 2 grades or something if you just watch all the videos. I
can tell you last year that students who used it, 50% of people who completed the course or went over 50% of the course went up. 50% of people went up 2 grades or more. 17% of people went up 3 grades or more. Some people went from a U to an A, and they went up basically the highest possible amount, where they didn't go U to A star. And maybe this year someone will go from U to an A star. Selena, you've been an absolute star. It was that was wonderful from you. I'm just turning your microphone off right now, so you are now muted. You can say what you can say. shit fucking bugger, whatever you want, because it's not going to be broadcast. Thanks so much for that. Okay, I'm going to cram through 2 more questions, and then I'm going to call it a day because I've done my hour and a half. I need to hand over to Richard, who's doing the chemistry course at 6 p.m. So you've got half an hour. Take a breath. Also, read the Facebook reviews, guys. Like, if you don't have to take my word for it. Selena's just told you what her opinions on it are. Like, go and have a look at those Facebook reviews. Whilst you're leaving your own Facebook review, you can see what they. Meiosis summary. How much am I per hour for tutoring? You don't want to know. Basically, you don't want to know. Literally, my course is cheaper than 1 hour of my time. You can buy the entire revision kit and get me to teach you all of AS, all of A2, all the practicals, the maths, the stats, all the exam technique is less than 1 hour as a private tutor if you want me. I'm expensive, but I get grades. So that's why people pay me that much. The essay question I'm going to cover in the essay web class. There's going to be a full web class on that. Rich, do you want another live Q&A? I've used the Ultimate Guide for a year, and Tom, I'll call you in another time. Chuck that in the chat so other people can see it. Mo's also there. Yeah, you can ask each other questions. There are plenty of people who use my courses in the chat, so ask them questions. So they are not going to be biased other than the fact that they like it because it's bloody awesome. Okay. Can you buy parts of the kit? Basically, no. You can buy the kit or you can buy the web classes. They're your choices. Who is the most active viewer? All the active viewer winners will be announced on Sunday at 6 o'clock in the web class. I'm going to download the list of the chat. I'm going to go through it, pick out some people who've asked them. I'm giving away those courses. They're all being given away at 6 p.m. on Sunday in the live web class there. Every web class is going to have a winner. So there's going to be, you can win. Every single web class is going to have an interactivity winner. So the biology, the chemistry, the maths, is 3 today that we're giving away. 2 tomorrow, 2 on Thursday, 2 on Friday. We're going to give away like 10 or 15 web classes. Sorry, 10 or 15 courses. The Instagram winner, the Facebook winner, all that's going to be given away on the web class on Sunday night. Application questions. Okay, so application questions, I'm gonna cover that a bit more in the exam techniques web class, so make sure you come join the other web class. The free web classes this week, I will cover some of that in them. The chemistry web class is today, starting in half an hour, so I need to hand over to Rich. One more biology question, and I'm gonna say goodbye. I'm not gonna do predictions right now. Meiosis summary. Okay, why are proteins only present on the why are proteins that are only present on one side of the membrane useful? Okay, so they may be associated with basically there's no content in the spec on this, so most of this is not that useful. So you might have a glycolipid or a glycoprotein, which is going to be a protein lodged into the one layer of the membrane, on the outer layer membrane usually, and it's going to have a carbohydrate attached, and they're going to be used for recognizing bits and pieces. So maybe you're going to have some like second messenger molecule binding to that, and it will mostly they're going to be transmembrane proteins. In terms of A-level biology, giving you an answer to that, basically you don't need to know. When can you get an AS prediction? Basically, all the content I covered today is the stuff that you have to know. If you want my solid predictions of answers that were on the screen today, I bet you'll get 15 to 20 marks of what I've been through today will be on the AS paper that you actually say paper 1. So my predictions essentially are the bits that are definitely going to definitely come up, and they were the things that were in today's web class. It's a pretty bit of a dangerous game. I might put together a bit of an educated guess for some of the predictions, and I will hold off on that until I've given it some more thought. I'm not just going to do it off the top of my head. But again, the stuff I did today is what's really you need to know. Okay. The recording will be posted exactly in the place where you click to join this live web class. It will be posted there as soon as it's ready, which will probably be this time tomorrow. So come back tomorrow. There's obviously web classes tonight. The recording will be posted in the course. The name of the lesson will change to recording. And if you want that unlimited unlimited access pass, don't forget the link. It's unlimited-free-trial. Thank you very much. See you later, everyone. Have a great evening. And